Hello everyone. Today again we are going to continue with biology chapter number 4, molecular basis of inheritance. In our last lecture we have studied the topic tRNA, the adapter molecule. We have studied about the role and functions of tRNA. Okay. Today we are going to study the topic translation. Okay, translation is also known as protein synthesis. Translation is also known as protein synthesis. Indirectly, we are going to study about the synthesis of protein. Okay, what do you mean by translation? We will study the definition. Translation is the process in which the sequence of nucleotide triplet. Okay, that means nucleotide triplet means codons that are present in messenger RNA. It gives rise to a specific sequence of amino acids during the synthesis of polypeptide chain or protein in ribosomes. So what is translation? It is the process in which the sequence of nucleotide triplet, that means codons that are present on mRNA, are translated and specific amino acid in a sequence is formed in the form of a polypeptide on ribosomes. Is it clear? I will repeat the definition. Sequence of nucleotide means codons that are present on mRNA are translated and specific amino acid in a sequence is formed into polypeptide, and this all things happens in ribosomes, on ribosomes. Okay, all type of proteins are synthesized by the cell within itself. That means this process is intracellular. All this process of protein synthesis takes place inside the cell and by the cell. Okay, and it is known as intracellular. Okay. Now, for this process of protein synthesis, what all things are required? We will see the requirements of protein synthesis. Okay, for the process of translation, we require mRNA, ribosomes, amino acids, tRNA, Mg+2 ions, amino-acyl tRNA synthetase, that means enzymes, and initiation, elongation and termination factors. Okay, all these are the requirements for the process of protein synthesis. One by one we will study about this components. Okay, first we are going to study about amino acids. Amino acids, we can say that it forms the raw material for protein synthesis. Amino acids are the raw material for protein synthesis. Total 20 different types of amino acids are known to form proteins. 20 different types of amino acids are required to form proteins, are known to form proteins. This all proteins are available in cytoplasm. Okay, what are amino acids? Amino acids are the raw material for protein synthesis. How many amino acids are there? 20 different types of amino acids are there which forms the proteins, and this all amino acids are available in cytoplasm. Next we will study the role of DNA in protein synthesis. What is the function of DNA in this process of protein synthesis? DNA performs the major function. It controls protein synthesis. Okay, DNA controls the process of protein synthesis, having amino acid in specific sequence. This control is possible through transcription of mRNA. Okay, we have already studied the process of transcription. How is this control possible? Because of the process of transcription of mRNA, DNA is able to control the protein synthesis. Genetic code is specific for particular amino acids. We have already studied this. A triplet code is there and a specific genetic code is present for a specific amino acids. So because of this property, DNA controls the process of protein synthesis. Next, we will study about the role of RNA in protein synthesis. RNA serve as an intermediate molecule between DNA and proteins. RNA is an intermediate molecule between DNA and protein. Okay. Next, and the major component of protein synthesis is ribosomes. Ribosomes forms the major component for the process of protein synthesis. It serves as site for protein synthesis. We can say that it is the site for protein synthesis. Each ribosome consists of larger and smaller units, 2 units. Okay, larger and smaller units. These subunits occur separately in cytoplasm. You can see here larger and smaller unit. One is 50S and the other one is 30S. Both of these subunits occur separately in cytoplasm. Okay. Only during protein synthesis, these 2 subunits get associated together due to Mg plus ions. Okay. Both these units are separately present in cytoplasm, but during the process of protein synthesis, both of these subunits get associated, means they come together, and this association is because of the presence of Mg plus 2 ions. Okay. Next is, a ribosome has binding sites. Okay. One binding site is present for mRNA and 3 binding sites are present for tRNA. Okay, 1 binding site for mRNA and 3 binding sites for tRNA. Those 3 sites are P site, you can see here P site, A site and E site. Both these large and small units are present. P site, A site, E site. P site means peptidyl site, peptidyl tRNA site. A site means aminoacyl tRNA site and E site means exit site. We can see here ribosome has 3 sites. A site, binding site for first aminoacylated tRNA. P site, binding site for peptidyl tRNA. And E site, binding site for the uncharged tRNA. These sites are present at the interface between small and large subunit of ribosomes. Okay, only first tRNA amino acid complex directly enters P site of the ribosome. First tRNA enters the P site of the ribosomes, and the next onwards, second, third, fourth, all those amino acid complex enters at A site. You have to remember this point because we are going to study the process of protein synthesis, and there we are going to see how this enters. So first amino acid complex enters at P site, and then all the amino acid complex enters at A site. Okay. Only first tRNA complex enters at P site of ribosomes. In eukaryotes, a groove is present between 2 subunits. These are the 2 subunits. In eukaryotic organism, a groove is present between 2 subunits of ribosome. It protects the polypeptide chain from action of cellular enzymes and mRNA from action of nuclease. Okay, I repeat. In eukaryotic organisms, a groove, you can see a groove is present in between the 2 subunits of ribosomes. It protects the polypeptide chain from action of cellular enzymes and mRNA from the action of nucleases. Okay. We are going to continue this topic in our next lecture. Okay. That is, we are going to study the mechanism of translation. Okay. This we have studied the introduction part, introduction of translation. and we have studied about the requirements of translation. What all things are required. Okay, and in that we have studied in detail about amino acids, DNA, RNA and ribosomes. Next time we are going to study about the mechanism of translation, that is the steps of translation: initiation, elongation and termination, and the post translation changes. Okay, thank you.